Hi everyone. Here we are, section 8.3, and our continued discussion about infectious disease. And here it's talking about emerging diseases and a little bit about COVID-19. Now, the concept of an emerging disease, it says an infectious disease can be an emerging disease if it's occurring for the first time in human populations, if it is rapidly increasing in its incidence or frequency in humans, or entering into new geographic regions. So it could be a brand new disease, or it could be a disease that's just becoming much more common, or one that's in an area that it's never been before. And we can give it the description of an emerging disease. And here's a world map showing you some of the emerging diseases. So diphtheria has been around a long time, but they apparently had a diphtheria outbreak, in 1993. Or here, I'm remembering in 1999 when West Nile virus was first found around New York City, and that was a disease that you only found in Africa. So it was an emerging disease in the U.S. Other ones in the U.S., the H1N1 influenza was the one we called swine flu in 2009. You might remember hearing about Zika virus when babies were being born with microcephaly, small heads. And, all these things, a lot of these have been around, but not necessarily in big numbers or in the area where they were found. So we call these emerging diseases. There's a little bit of discussion here about identifying pathogens having the potential to be emerging diseases. And so there are organizations that keep track of these kind of things. One of them might be a little bit surprising. Here's Streptococcus. It says emerging or re-emerging diseases. So it might be that the disease declined and then it's coming back. So that's another variation on that. Here it says Streptococcus, which is the bacterium that causes strep throat and other infections, is considered a re-emerging pathogen due to its increased resistance to antibiotics. So we're getting strains of Streptococcus that are actually acting differently than they have in the past because they've had mutations that allow them to become antibiotic resistant. So we would consider that an emerging disease. Here's just a little discussion that the World Health Organization, the CDC, Centers for Disease Control, are organizations, major organizations in the world to keep track of these kind of things. So there are people that are looking at this and keeping track of what diseases are around. A little discussion about COVID-19. Here's the virus. So COVID-19 is the disease that we get, and SARS-CoV-2 is actually the virus that causes COVID-19. And this is what we call a coronavirus. And I mentioned that before because we said the coronaviruses have lots of spikes on the surface that kind of looks like a crown. So they call them coronaviruses. And so you should be well aware that SARS-CoV-2 is an emerging disease in all over the world right now. And so it's caused a pandemic, but it also is emerging because it's a new virus. And it says, let's see, the coronaviruses are well-known viruses. They've been around for a long time. Here's one that said it was first recorded in the 1950s in humans, although we've known about coronaviruses in animals for longer than that. But in that time, coronaviruses were just causing the common cold. That being said, coronaviruses have been on the radar of scientists for a long time, knowing that they could mutate and transfer from animals to humans. And so you can see in some of this discussion here that it's believed that coronaviruses have jumped from animals in multiple times to cause human disease. Now, you might be a little too young to remember this, but there have been a couple serious virus outbreaks or emerging diseases in the coronavirus family. And the virus SARS-CoV, now remember, COVID-19 is SARS-CoV-2, but there was this one called SARS-CoV, or we just called it was in the news, we just called it SARS. Broke out it emerged in, first recorded in China in 2002. Thought to originate from bats or civet cats and transmitted to humans. And there was a global outbreak, although it says the outbreak totaled over 8,000 cases and 774 deaths, with the fatality rate of just under 10%. And that's the kind of thing we were really worried about with SARS-CoV-2, that I think SARS-CoV-2 has a 1% or somewhere around that death rate, but this one has a 10%, and that would be a much bigger deal. And so when SARS-CoV started spreading, there was a lot of activity to try to squelch it as quickly as we could. The other coronavirus that has been a problem with humans was the MERS. So MERS is similar to SARS, but again, it's another coronavirus like COVID-19, like SARS-CoV-2. And it says it appeared in Jordan in 2012, so it got a Middle Eastern respiratory virus. The MERS was one that when it started infecting people, the action that people took was really dramatic because it actually has a fatality rate of 34%. That's, again, a game changer. I think a lot more people would be concerned about our current pandemic if the death rate was that high. Think about that. Think of all the people that you know that have gotten that, and with a 34% death rate. So when MERS was around, it was only around for a short period of time because it really got squelched quickly. And it probably had something else to do with the virus, why the virus didn't propagate really well. But there still were 858 deaths. I think the closest physically that it got to New York State, I believe that there was an apartment complex in Toronto where some people were being kind of, they claimed being held hostage, but they were quarantined and not allowed to leave uh, because there was MERS there. And I think the whole apartment complex got shut down. They were all quarantined. So it can be a really big deal. And coronaviruses are known to mutate a lot. Here's a picture of that SARS-CoV-2 virus, and this is the one I said kind of looks like a crown. It has a RNA for its nucleic acid, a capsid underneath, and what you see here is the envelope and the spikes. There's more information there, and yeah, there's some questions here that you can answer. So I'm going to go to the next section on antibiotic resistance, section 8.4. And this is our last topic for this unit. And what we want to talk about is just a little bit about antibiotic resistance. So probably a lot of you have heard of this, the concept of antibiotic resistance. And it's the idea that when populations of bacteria are being controlled with antibiotics, so antibiotics are drugs that kill bacteria, when populations of bacteria are being controlled by using antibiotics, some cells in that population could have some resistance to the antibiotic, and the use of the antibiotic can turn the population to be primarily antibiotic resistant. Now, in lab, I do a little discussion about evolution and natural selection, and that's what you have to understand to have a good understanding of how a population becomes antibiotic resistant. But what they're showing you right here, this is would represent like the way we would grow bacteria in the laboratory, that we would grow them on an agar plate, and you'll see that in the lab. And you have all different, here, this is one kind of bacteria, and they're all growing on the plate. Now, what happens in any population is you're going to have some genetic variation. You're going to have some variation. Think about any group of people. You look around and everybody looks a little bit different. And that's because we have genetic variation. If you're in the same family, you look a little bit more alike than if you're not in the same family. And so that's because you have more similar genes. So this is just showing you a cartoon of a population of bacteria, and most of them are sensitive to a certain drug. They'll die if they're exposed to a certain antibiotic. But there's going to be some variation in the population, and maybe a couple of the cells actually got a mutation that made them resistant to the drug that you're using. So this is a normal population of bacteria in any environment, that you're going to have a mixture of sensitive and resistant ones. Now, once you expose that that plate or those organisms to a drug, usually an antibiotic, that drug will be able to kill most of the bacteria that are sensitive to the drug. And so this is showing you the antibiotic killing the cells. And then when you're done, the only cells that are going to be left are ones that are resistant. The antibiotic resistant cells survive, and the sensitive ones will die. And so what's going to happen is you're going to change your population from being made of primarily sensitive cells to primarily resistant cells. So you're going to kill off the ones that are sensitive and leave room for the ones that are fitter, that have greater fitness, the ones that can survive the antibiotic. And your population is going to become primarily antibiotic resistant. And so this is what's happening to create populations of antibiotic resistant bacteria. You might remember in evolution, and I talk about this in lab, that we have natural selection. So you're going to be killing off the antibiotic is used as an environment that's going to create natural selection by killing off the sensitive ones and allowing the resistant ones to grow. So our population then is going to become primarily resistant bacteria. And now we've got a more likely to have a population that resists penicillin or streptomycin or whatever antibiotic that is. There's a lot of bacteria that are becoming highly antibiotic resistant. We used to have, most of them were sensitive to antibiotics to the point that most of them that we come across in clinical situations are resistant to antibiotics. And some of these infections are those that microbes that cause tuberculosis, malaria, gonorrhea, the sexually transmitted disease, Staphylococcus aureus, when somebody has a staph infection, it's caused by this organism. And so there's a lot of different organisms that we're finding more and more antibiotic resistant organisms. It talks a little bit about why we're having a hard time with this. Well, one is it's just the nature of biology. Evolution and natural selection happen. And so that's what you're seeing here. By using antibiotics, we're selecting for antibiotic resistant organisms. Also, it's a very complicated and challenging process and expensive process to develop new antibiotics. So it's something that pharmaceutical companies are reluctant to do because it's a lot of research and it's not easy. And on top of that, it would be a drug that wouldn't be used a lot, hopefully infrequently. A lot of the pharmaceutical companies would rather spend their money making drugs that people will use every day for the rest of their life, like high cholesterol drugs or diabetes drugs, things like that. So how do we avoid making antibiotic resistant organisms? One is, if you do take an antibiotic, to take all your whole prescription. Don't take part of it, because what you're going to do is you're going to select for the ones that are even slightly antibiotic resistant, and they're going to be common in the population. Don't always expect to get an antibiotic. Antibiotics do not work against viruses. They only work against bacteria. And don't save antibiotics to use them again later. You really want to only use them when prescribed by a doctor. So what's happening with antibiotic resistance? Well, we have the development of these multi-drug resistant organisms. So we used to be able to treat, for example, tuberculosis with streptomycin, but most tuberculosis is resistant to that now, and we're getting, and so other drugs have been tried. But what's happening is we're getting these multi-drug resistant organisms, MDRO, multi-drugs. So these are organisms, bacteria, that are resistant to more than one kind of antibiotic. Now, some of the terminology you see, XDR tuberculosis, is something we're very concerned about. And that means extensively drug-resistant tuberculosis. That here we have, down here it mentions multi-drug resistant tuberculosis. Multi-drug resistant tuberculosis isn't as bad as XDR. XDR is even more resistant, and it's resistant to almost all the drugs that are available. Um, the it says the extensively drug resistance still pretty rare, only 63 cases in the US since 1993 to 2011. Multi-drug resistant is much more common. And so usually when people have tuberculosis and they're being treated, they can be treated by their antibiotics for months, like 6 months. And it's a really big deal. I mean, it's really hard to get rid of this. Another pathogen that's becoming very common is MRSA, and I know a lot of you have heard of MRSA. What it means is methicillin-resistant Staphylococcus aureus. So again, you've heard you get a wound and you can get a staph infection, or sometimes you just get an infected blemish on your face or something, and it can be a staph infection. And we used to just give, penicillin, and it would be fine. But now the of um, a lot of staphylococcus is resistant to penicillin, amoxicillin, and now even becoming resistant to methicillin, which was designed to fight staph. So it even says here, boils or staph infections. This one, I've actually experienced this statistic. It said in 1974, only 2% of staph infections were caused by MRSA, the drug-resistant staph. But by 2004, the number rose to 63%. And I started teaching at Corning Community College in 1998, and I never heard anybody mention MRSA. And then it was kind of the early 2000s, I started hearing people. I remember like the first couple times I heard people that knew someone who had a MRSA infection, or I remember even finally meeting a student in my class who had a MRSA infection, and it was very unusual. And now it's very common. I hear about it on a regular basis. You know, being a microbiology teacher, a lot of students will tell me about interesting infections and so I hear about this all the time, to the point at first I was just shocked when I heard about it, to the point now it's kind of a relatively common thing. But you don't want to, you get MRSA. It doesn't mean you're going to get any sicker. It just means it's going to be harder to treat it and get rid of it. And that could be a problem. Now, the hospitals have really put a lot of effort into reducing microbes in their environments, and infection rates, it said, have declined dramatically in hospitals since we've been doing a lot more work to get rid of MRSA. Here it says also problem in nursing homes. And I think that's where I'm going to end on antibiotic resistance. I don't think I put these on my, I think I forgot to put the check your progress on the handout, but I would say all of those would be relevant for you to review. So there you go. That's the end of week 12 material. Take care.